All right, so we're now going to move on to talking about meiosis. And meiosis is going to be involved with sexual reproduction. Okay, and so in this, what sexual reproduction is producing, it produces offspring that contain a unique combination of genes from mom and dad. Okay, so like before, we're going to start talking about, homologous chromosomes. So you always are going to have one from mom and one from dad. Okay, and I like to draw them in different colors because I recognize that while they are the same chromosome, they are different. You got one from your mom and one from your dad. And so they can potentially have different kind of alleles, which is a term that we'll learn soon, which are versions of genes. Okay, now when we start talking about meiosis, we're going to start talking about specific types of cells, but I wanted to mention briefly kind of the typical body cell. So typical body cell. Okay, and another word for body cell is somatic. So these are kind of body cells, typical ones. And so if we took your cheek cell, or we took a skin cell, or we took, a piece from your liver, those cells are going to be diploid. Okay, so they're diploid, which means that they have two copies of each chromosome. Okay, and if you remember, we have 23 chromosome types, and we say there's 2 of them, one from mom, one from dad, which means that there are 46 chromosomes in each somatic cell. Okay? So each cell, if we look at that, we go through and we could see you have a chromosome 1 from your mom, a chromosome 1 from your dad, a chromosome 2 from your mom, chromosome 2 from your dad, and so on and so forth. To look at your actual chromosomes, and you can do this, okay? So when we kind of look at an individual's, all of their chromosomes, so we have this term called a karyotype. Okay, and so this is going to be kind of the chromosomes of an individual. Okay. So I wanted to show you what this actually looks like. So this is what a karyotype would look like. So this is an individual, and this is a human. So there are going to be 23 sets of chromosomes. So here's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and this last group, which is 23. And so I'm going to talk about this group 23 right here because it's a little bit different. What we're actually looking at is that each of these is they're homologous chromosomes, okay? So this is maybe from mom, this one's from dad, so we could say mom and dad. And then each of these is then going to have a sister chromatid attached to them. Okay, so remember when we go through mitosis, there's a point where you end up having 2 of each of, the individual chromosomes. Okay, so these are sister chromatids to each other. These are sister chromatids, and then to each other, they are going to be homologous chromosomes. The reason why there's 4 at this stage is because when they do an actual karyotype of an individual, they take the cells at metaphase and they pull them out, and at that point you would then have, it's prior to them being separated from each other. So the sister chromatids are still attached. Okay, so a little bit more about the actual chromosomes. So for humans, again, we have 23 chromosomes. Okay, and we're gonna have 2 of each for total of 46. Okay, and again, The 2 of each is one from mom and dad. All right, so 1 through 22 of the chromosomes are what we consider autosomes. Okay, and autosomes mean that the chromosomes, so the one from mom and one from dad, are matching. Chromosomes match. So they are, you know, you have chromosome, you can just draw this out. So we have chromosome 1 from dad, chromosome 1 from mom. And let's just say the chromosome 2 is slightly smaller. So we have chromosome 2 from mom, chromosome 2 from dad. But they're identical to each other, so they're a match. The 23rd, okay, because we have 23 chromosomes, is called a sex chromosome. Okay, and these do not necessarily match because you can have XX or you can have XY. Okay, so the sex chromosomes are an X chromosome and a Y chromosome, and depending on the combination that you can either get an XX or an XY. You will then be chromosomally female or chromosomally male. Okay, and so if we're looking at this karyotype, this individual, it has an X chromosome and a Y chromosome, so this would be a male individual. Okay, and so again, I want to show that these are all the autosomes, so they all have matching, they're the same height, everything's identical, except for maybe the versions of the genes, but they're all going to have the same genes on there. Okay, so when we go through the process of meiosis, we're going to have to pay attention to kind of now these homologous chromosomes and the sister chromatids as well. Okay, so again, 23 chromosomes in all. There are 2 of each one. So 1 through 22 are called autosomes. So they're same, you know, same actual size, same genes, everything's the same. Okay, when we talk about the sex chromosomes are not identical to each other. The X chromosome has a lot of genes on it. The Y chromosome mainly has genes for becoming male. Okay, so it has to do with like testes, testicle development, things like that. Okay, so they do not have the same genes on them. Okay, they're different sizes. The X chromosome is often very large, and you see the Y chromosome is usually pretty small. And so they are considered the sex chromosomes. They're not autosomes because they don't match up necessarily. Okay, if you're a female, yes, they will be the same size. Okay, and when we're talking about, again, we've mentioned this before, so I really want to make sure this is very clear. When you have kind of the copies, one from mom, one from dad, they're the same. And I know I drew them a little bit different size, but they're the same. These are going to be considered homologous chromosomes. Okay, and so they carry genes controlling the same inherited traits. Okay, so like eye color or hair texture, things like that. All right, so when we're talking about meiosis, we have to start talking about gametes because we are not going to be doing the same process of mitosis. Okay, so we're going to say gametes and the life cycle of a sexual organism. Okay, so this is going to be kind of the life cycle of a multicellular organism. Okay, fertilization is going to be part of this. Okay, so fertilization to produce its own offspring. Okay, actually I should say production. Okay, so this process is the life cycle from fertilization until production of its own offspring, okay? So what I wanted to kind of go through right now is kind of just a cartoon version of this. Okay, and so these are gonna be gametes. Okay, and so we're all gonna start off, you know, we have egg, it's gonna have 23 chromosomes, and I'm gonna talk about how we get to that 23 chromosomes. Sperm are gonna have 23 chromosomes. They're going to go through the process of fertilization where the sperm is actually going to penetrate the egg and is going to actually inject its DNA into the egg. Okay, and so we're going to call this fertilization. Once that happens, we then have what we call a zygote. And because we now have 23 chromosomes from mom, from this egg, and 23 from dad, we are now going to have 46. So that's 23 from dad plus 23 from mom equals 46 total chromosomes, okay? The zygote will develop into an adult. And if this is a male adult or a female adult, then, so let's just say over here we have a male adult, they will then ultimately start to produce sperm. Females will produce eggs, and this process can start over. So we're talking about the process from fertilization, so from when this unique individual comes together to form a zygote. So this is a human, or we could say a baby, until that zygote ultimately develops into a human adult that's in puberty that can then start to produce male or female, depending on what kind of individual we have, okay? So what's happening is we need to talk about how to make cells with only 23 chromosomes, because in our the previous, when we talked about mitosis, what happened is that we started off with a cell that had 46 chromosomes. We doubled the DNA and we ended up with cells with 46 chromosomes at the end. So 46 and 46. For meiosis, we don't want that to happen. We're gonna start with a cell that is 46, okay? And actually is going to replicate, it's gonna have even more chromosomes to that, but this cell is going to ultimately need to end up, and I'm gonna do this, is gonna be kind of confusing for a second, but then those cells are ultimately going to turn into we're gonna end up producing 4 cells that are gonna have half the number of chromosomes. Okay, and so this is actually, I should put 92 because we're gonna talk about this in a second. All right, so meiosis is going to allow for a reduction in the number of chromosomes. And the way that this has happened is that there's actually 2 divisions. Okay, so in mitosis, we have 1 division. In meiosis, we're gonna have 2 sets of divisions where we're gonna go from 92 chromosomes to 46, 46, and those are gonna be split off into 23, 23, 23, 23. Okay, so I wanted to show this a little before so we can kind of talk about what's gonna happen. So we talked about how, you know, at fertilization, we have a zygote that's ultimately gonna turn into an adult. The male will then produce sperm when he hits puberty, and the female will start to produce eggs. Those then can come together and to produce a new organism. So what we're gonna have happen in meiosis is we're gonna take cells that originally are gonna have kind of 2 copies of the chromosomes and ultimately produce haploid gametes, which have 1 copy of each chromosome. Now, the reason why we have to do this is because if we have sperm cells with 46 chromosomes and egg cells with 46 chromosomes, if they come together, they're gonna produce a zygote with 92 chromosomes, which is way too many. This is not a normal human. Normal humans have 46. Okay, and so we're gonna talk about how we actually do a reduction in the number of chromosomes. Okay, so now we're gonna talk about meiosis in more actual detail of how this is going to take place. So I'm going to start drawing my chromosomes in colors. Okay, and so we're going to start off with what we've seen before. So we can have a chromosome from mom and a chromosome from dad. Okay, now just like in mitosis, we're going to have DNA replication take place. Okay, and in that process, what's going to happen is that each of the homologous chromosomes is going to have a sister chromatid that's going to form. Okay, so what you're going to see is now at this point we have 2 of each chromosome. Okay, and so if we have 2 of each chromosome, there's 23 chromosomes. So again, that's 46. At this point, we're actually going to have 4 of each chromosome, which is going to be 92. Okay, so this is going to be the number of chromosomes we're actually going to start with. I'm only showing one set of chromosomes, but you'd have to imagine that there are 22 other chromosomes that are not drawn. So in the first step of meiosis, we're going to call this meiosis 1. We're going to split homologous chromosomes apart. Okay, and so what's going to happen is we're going to have the blue are going to separate from the orange. Okay, so this is going to be meiosis 1. Okay, so we have homologous chromosomes are going to split. We're then going to have another division that's going to take place, and we're going to call this meiosis 2. Okay, and what happens in this is that we're going to split sister chromatids. Okay, and so you're ultimately going to end up with something that looks like this. Okay, and again, I'm not drawing all of the different chromosomes, but like if I was to put these in their cells, we're going to end up with 4 cells at the end. Okay, because we start off with 1 cell. Okay, after replication, then those cells are going to divide where we have the homologous chromosome split, and then we're going to do meiosis 2, which is when we're going to split the sister chromatids apart. Okay, and so this is different from how we actually have mitosis take place. Now, within each of the steps of meiosis, we actually have what we call the similar stages. So there's going to be a prophase 1, metaphase 1, anaphase 1, telophase, cytokinesis 1 for meiosis. And then there's going to be a meiosis 2, which is going to have a prophase 2, metaphase 2, anaphase 2, and telophase. Okay, and so I'll kind of show how that is going to work. Okay, so here is just a kind of cartoon version of that. So we're going to start off with, chromosomes, 1 from mom, 1 from dad. They're then going to be duplicated so that we have, at this point, we have like 4 of each chromosome. Okay, so we have 2 of these sister chromatids, 2 of these sister chromatids. They're then going to divide. Okay, sister chromatids or homologous chromosomes are going to split during meiosis 1, and then in meiosis 2, we're going to have sister chromatids split. Okay, and so what I want to do right now before I get into a little more detail about how this is going to happen is I want to start kind of getting a tally of how mitosis and meiosis are different from each other, because that's one of the big things I like to kind of test on is, so for mitosis, we're going to have just 1 division. Okay, so we talked about that. Mitosis just has 1 division. This is going to have 2 divisions. Okay, so we have meiosis 1, meiosis 2. We're going to start with 1 cell. So start with 1 cell, and we're going to end up with 2 cells in mitosis. In meiosis, we're going to start with 1 cell, and we're going to end with 4 cells. Okay, because we're going to go through the process twice. So we have one cell going to two cells, and then those are going to split up. So we ultimately start with one, we end up with 4. When we do this one cell to two cells, these are going to be identical to each other. Okay, so all the same chromosome, everything is identical. These are going to ultimately be non-identical. Okay, and so if I show you this, some of these have red chromosomes, some of them have blue chromosomes, so they are not identical to each other. So non-identical, which is important because that's what we want to happen in sexual reproduction. These are going to have kind of end with 46 chromosomes. Okay, so they start with 46, end with 46. These are going to end with 23 chromosomes. Okay, so we can say these ones are diploid. These are going to be haploid. All right. Now, what I just showed you is right here is not the whole story. So we're going to have a couple other things that can actually take place to then create even more diversity in the ending 4 cells. Okay, and so this first type of kind of additional variation is going to be coming from something called crossing over. So let me start a new sheet. Okay, and so crossing over is going to be kind of a way that homologous chromosomes will basically swap parts of their chromosomes. Okay, this is going to all happen during prophase 1 of meiosis. So what's going to happen, and so we've seen this before, I'm going to draw, we've had DNA replication already take place. And so what's going to happen in crossing over is we're going to actually have parts of the chromosomes switch places. Okay, and so this is called crossing over. So I'm going to only show part of crossing over taking place. So what's going to happen is whole pieces of the chromosomes are going to switch places. Whole pieces are going to switch. Okay, and so what you're seeing here now is what we call recombinant DNA. So this has been recombined. So if we were originally saying this is from mom, this is from dad, we now have chromosomes that have part mom DNA and part dad DNA. So these are recombinant DNA. Okay, and so how this would take, kind of what this would then look like in our actual mitosis, our meiosis. So let me just draw this out. Okay, so we're gonna have, so I'll draw it like, I'm gonna draw it a slightly different direction. Makes it a little bit easier to see. So I'm gonna draw them sideways. So we would have kind of duplication of our chromosomes. So we have sister chromatids. Okay, and then in prophase, we're gonna have crossing over take place. Okay, and so I'm gonna draw something similar to what we've seen. And crossing over always takes place. So there's not like it sometimes happens. So what's gonna happen is that then this would go through kind of meiosis 1. Okay, and so we're gonna end up with a cell right here and a cell right here. This one is going to have kind of a combination of the blue and the orange. Same with this one. Okay, and then we're gonna go through meiosis 2. Okay, and so we're going to end up with 4 cells. This one is going to have okay, and so what I want you to notice is that these are all completely different from each other. You have one that has kind of a full orange. This one has both dad and mom DNA. This one is going to have both mom and dad DNA, and this one is going to have just, I think, mom DNA. Okay, and so this is what crossing over looks like. Okay, so we're having the actual swapping of parts of the actual chromosomes. Okay, and this adds genetic diversity to the gametes. So if these are eggs or sperm, they're going to have kind of different actual combinations. Now, an additional way of doing recombination is we're going to do something called independent assortment. Okay, and so when we talk about independent assortment, this is basically how the chromosomes line up in metaphase. Okay, so when chromosomes line up in metaphase, they do that independently. Okay, and the only way to really explain this is to actually show you how this works. So let me start drawing a new one. Okay, and when we start talking about independent assortment, I actually have to start to talk about more than one set of chromosomes. So for this case, we're going to end up having kind of a big chromosome. And I'm not going to show crossing over just to save me a little bit of time to do this. But basically, what can happen is that you could line up, this is how these chromosomes are lining up during metaphase. Because now you're going to actually have separation of the homologous chromosomes, so they all stay together. But depending on how things line up, we could have the blue on this side, on the right side, and the orange are going to be on the left side right here. Okay, so that's one possibility. Or it could be, we keep them on the same side. Okay, and we'll say a little orange down here. But depending on how they line up, it's going to make it so that our kind of different cells at the end are going to be different. Okay, and so if we stick with this, it's going to be very similar to what we see in this situation where, they're all going to end up on the same side. But if we have this independent assortment, so the orange actually line up on this side, what's going to happen is that during meiosis 1, we're going to end up with kind of a blue big set and the orange little set. And on this one, we're going to end up with the orange big set and the little set. Okay, and then ultimately at the end, we're going to end up with 4 cells. So we can have a big blue, little orange. This is going to have a big orange. This one's going to have a big orange, and then a little blue. Okay, and so what you're going to see is that you'll have different combinations. That's not what would have happened if we had this group. If we stick with this, at the end, the 4 different cells, and I'm just going to draw these quickly, you would have one big blue, one little blue, one big blue, one little blue, big orange, little orange, big orange, little orange. Okay, so you can see that these are different from each other. So these are different types of ways that they can line up, which is called independent assortment. Okay, so it's random. The way that they line up is completely random. As long as they line up with their homologous chromosome and sister chromatids, It doesn't matter if you have blue on one side or orange on the other. It's going to kind of add to this variation. Okay, so I want to go through kind of the actual steps of meiosis 1 and meiosis 2. And again, like I mentioned before, we call them the same things as mitosis. So in prophase 1, this is when this one's not showing the same exact stuff. It's only showing 2 sets of chromosomes, but you'd have crossing over take place. Spindle fibers are starting to form. You have the sister chromatids still stuck to each other. Homologous chromosomes are lined up. In metaphase 1, you have your pairs of homologous chromosomes lined up to each other. There's independent assortment, so it could have blue on this side or blue on this side, red and red, as long as the little ones and the big ones are lined up together. So metaphase, they line up in the middle. Anaphase, they're going to pull apart. And in this case, we're pulling apart homologous chromosomes. At the end, telophase 1 and cytokinesis, we're going to end up with 2 cells that each have kind of 2 copies of each chromosome, which are going to be those sister chromatids. When we go to meiosis 2, this is basically mitosis. Okay, so you have, 2 copies of each chromosome. In prophase 1, you have the nucleus is going to degrade. You have the spindle fibers forming, but we're having this happen in 2 different cells now. In metaphase 2, they line up in the middle. In anaphase 2, they're going to pull apart the sister chromatids. And at the end of telophase 2 and cytokinesis, we end up with 4 non-identical haploid cells. Okay, so they have 1 of each copy of the chromosome. So we no longer have 2 copies of each chromosome. Okay, so that is quite different from the actual process of mitosis in the fact that we end up with 4 non-identical. Okay, and so I just wanted to kind of, again, be able to compare and contrast mitosis and meiosis. So if this is our parent cell for mitosis, we're not doing all the individual stages, but you're going to ultimately duplicate all of your chromosomes. They're going to line up in the middle and they're going to separate, and you're going to end up with identical cells. So if you look at kind of what you started with and what you end with, they are identical. Okay, so you have, big blue, little red, big red, and you're going to have them here. So in mitosis, you start with one thing and you're going to end up with 2 identical versions of that. Okay, so that's mitosis. In meiosis, it's a little bit more complicated, where you are going to have crossing over take place. You're going to have homologous chromosomes align and then separate. And then you're going to go through another actual division. So 2 divisions. So here's division 1. Okay, and then these 2 are then ultimately going to divide again, and you end up with 4 non-identical. Okay, so if we look at the parent cell, okay, so parent cell has, 1 big blue, 1 little red, 1 big red. These have different combinations. So this one has a big red and a little red, but it's not even completely red. So these are kind of haploid versions. These are going to be the gametes. All right. All right, and then here is just another kind of diagram of how we have independent assortment. So these are going to be just probability. There's not one that's going to happen more likely than the other, but you have how your homologous chromosomes are actually lining up. So you can have red on one side and blue on the other, or you can have a red on one side, blue right here, and then blue and red. And so how they're going to divide then will then create these different combinations of the sperm and the egg. So if these are gametes, you can have lots of different varieties. Now, the big thing, because of the independent assortment, what I want to kind of mention is that for a human, we have 23 chromosomes, but we have 2 of each one. So that's 2 to the 23rd. Okay, so we have 2 versions of each one. Okay, and kind of depending on how they line up in meiosis, you can potentially have 8 million possible chromosome combinations. Okay, so, maybe for your first chromosome, it goes, they can go into one sperm and one can go into the other sperm, but then, chromosome 2, you can have all these different variation. So the main thing is just to know, I'm not going to ask you about this number, but basically a single man and a single woman can produce a zygote, or zygotes can produce zygotes with 64 trillion combinations of chromosomes. Okay, and all of that genetic variation is coming from the fact that there are 2 of each copy. You have one from mom, one from dad. And so what you give on to your next generation, it could be, lots of different combinations. And so crossing over is going to increase this variation. So crossing over, and also independent assortment will also cause that. All right. To end up, I wanted to talk briefly about, we talked about what goes wrong with mitosis. Well, some things can go wrong with meiosis. Okay, so this is a meiosis error. Okay, and this is going to be called non-disjunction. Okay, and basically what happens is that there is an error in meiosis. And basically what's happening is that the chromosomes are not splitting correctly. Are not splitting correctly. Okay, this can happen in either anaphase 1 or anaphase 2 in meiosis. So those are just the 2 steps when you're actually having division of the cells or of the chromosomes. So this is what could actually happen. So what you can end up with is having abnormal meiosis take place, and then let's just say we have an abnormal egg. So this one has 2 of this one chromosome right here when there should only be 1 of them. Okay, because at the end of meiosis, you should have just 1 copy of each chromosome. They have the correct number of this little chromosome. Here is a normal sperm, so it has the correct number of this big chromosome. But when they come together, what happens is that you end up having 3 chromosomes of the same type. Okay, and that's too many. So this is going to cause an abnormality. Some kind of common abnormalities you probably have heard of. So one of them is called a trisomy 21. Okay, and this is Down syndrome. Okay, and so what happens is that the chromosomes do not split correctly, and you end up having 3 chromosomes 21. Okay, so you have chromosome 21, 3 of them, 3 copies of it. Okay, and so let's look at, it's kind of hard to draw this version, so I'm going to actually just show you the picture, but this is how non-disjunction takes place. So there's 2 different types that can happen. You can have one that happens in meiosis 1, and then this one's going to be happening in meiosis 2. So for meiosis 1, what happens is that in the first division, 2 of the homologous chromosomes fail to separate. And so all of these large chromosomes are going to go into one of the cells. So over here, you end up having not the correct number. So there's actually 0 of the large chromosome here. And on this one, there are way too many. So there should not be this many chromosomes here. What that's going to end up doing is creating 4 abnormal gametes. So you have 2 gametes that are missing a chromosome, and 2 gametes that have an additional chromosome that they don't need. Okay, so these are going to cause issues. When we have meiosis 2 non-disjunction, so the beginning starts normally. So you have splitting of the homologous chromosomes, so each of them ends up with the correct number. But when you go through the second anaphase, you have sister chromatids that fail to form. So in this case, you actually end up having some abnormal eggs or sperm. So these ones are completely normal, but then these ones are not, because this one doesn't have enough chromosomes and this one has too many of this large chromosome. Okay, and so that's kind of how this process takes place. So if the chromosome that they add with an additional one is chromosome 21, that would then mean that that person could potentially have Down syndrome. So Down syndrome is a really common one. So again, 3 chromosome 21s. And there's a couple other types that are more common. And so these are abnormal numbers of sex chromosomes. Okay, and so the normal for female is XX, the normal for male is XY, but there are a couple of times where you can have XXY and XO, and this actually means that there is no, there's no X or Y, so there's actually just, they only have one X. So this is called Klinefelter's syndrome. And this is called Turner's syndrome. Turner's syndrome. So in both of these cases, the individuals kind of have a combination of masculine and feminine kind of stereotypical traits. They're usually sterile, which means that they cannot produce viable sperm or egg and will not be able to have their own children. Okay, so that's kind of some things that can happen with meiosis.